The project title that we have been doing is integrating genetic, agronomic, and economic approach to improving environmental adaptability and end-use quality of creeping red fescue. So two of my collaborating colleagues, Calvin Yoder and Talon Gator, have contributed to this presentation. First of all, I would like to show you general picture of weather here for the last 3 years compared to 30 years average. And you see these green bars, different levels in different years that shows different level of precipitation. And again, with those uh, line graphs showing uh, maximum, average, and minimum temperature, that shows that the weather condition year to year is pretty variable here at Beaver Lodge in Alberta. So I'd like to show you baseline soil nutrient status. And so what you see here is the nutrient levels at different soil profiles. They decrease along the depth, but the first top layer is most important for agronomically for plant growth. So if you see first top layer, the nutrient status seems to be quite in optimal range. So our project objectives, we have 3 sets of objective. One is breeding related, and that aims to improve population of creeping red fescue, the turf grass species we've been working with. And second component is improving its agronomy cultural practices through plant nutrition, growth regulation, and stand health management. And third objectives is determining economic profitability of different crop management factors included in the study. And 2 of those objectives are actually activities are in progress to meet those. And third objectives will uh, complete it once we'll have all agronomic information in hand. So what we had planned in 2020-21 and what we accomplished is in the breeding aspect, we selected about 7% healthy plants from 4 different populations. And we also tested whether it is possible to produce seeds in the greenhouse. And for that, we did both indoor vernalization and outdoor vernalization by putting plants outdoor over winter and bringing them in the greenhouse. But there was no reproductive success. And in agronomy side, we conducted 3 sets of on-station field experiments, 3 sets of on-farm, that is farmers field experiments, and some seed yield and quality assessments. So in objective 2, yeah, and so actually we are not able to collect all variable we intended in 2020. That's why we submitted a change request for following years, and that has been already approved. So let's go for these creeping red fescue breeding nursery, little bit photographs here in 2020, 2019 and 2020. And so more or less at different similar stages of crops. So this is a photograph of a greenhouse vernalization test we did. And in one of the tests, first test, we exposed plants to cold 4 degrees Celsius condition. And we put plants for 2 to 20 weeks in that condition and to see how many weeks of vernalization can be effective for reproductive success. And we did not have any success. And second test, I explained earlier, we vernalize plants over winter in outside condition and transfer them in February to the greenhouse, and still the results is no reproductive success. There was excessive tillering in plants, and probably plants ran out of resource, or there may, yeah, we saw actually lots of root boundness there. So it may be because of inadequate light, or not enough um, space for rooting, we are not able to have a reproductive success. So this is a result of plant selection. We selected about 7% most healthy and more number of heads producing plants. Based on that, we tried like how much is the difference uh, in seed yield uh, compared to the average of the population. And we saw most variability in boreal population with 197% selection differential. And these BRF1, BRF2, and Oracle are actually descendants of boreal. That's why we did not see as much variability, and hence the selection differential is little bit less than that of boreal. And this photograph in the right side, you see the plants infected with stem eye spot. That's what it is called, and caused by Didymalia fescue. And in our selection, we try to avoid this disease as much as possible, because it may cause up to 70% or more yield loss. So for our coming season plan, actually, since we did not have reproductive success in greenhouse, and we transplanted those clones in the field to have seed production in the field. But we need isolated condition for that, and we are planning on having high tunnels to cover them to avoid cross-pollination between the populations. Next, this is a agronomic experiment, and I have taken photographs from different 3 sets of experiments. And so this 2019 first production year, 2020, that means different set of trials. And this is third set is different set again with different sets of treatments. And I'll show you the results of them, what we saw. So here are 9 different treatments, and details of them are here. Down here, what you see is different nutrient application, and starting from one check, no nutrient applied uh, externally, and other nutrients are nitrogen, phosphorus, potassium, sulfur, and foliar application of boron. Then uh, uh, fungicide applied on the crop to have fall tillering health, and plant growth regulator application, and stacking those treatments one after another, and detail is given below. And for nutrient application, we had 2 methods, fall broadcast and fall banding. So even with these all various treatments, we did not see significant difference in seed yield over 2 years. And from the experiment established in 2018, that the first year results was in 2019, and second year results from the same stand 2020. Similar result from 2019 established experiment, and same set of treatments we had, and still there is some difference we can see here, but those are not statistically significant. So in terms of seed quality, if our treatments had any effect on seed quality, and we measured 1,000 seed weight, and we also did germination test, and there was also no significant difference of significant effects of the treatments. So the story is a little bit different of on-farm testing results. And so I'm showing from 3 different sites, and 2 of them are similar sets of 5 treatments. And there we applied plant growth regulator trinexapac ethyl and chlorum quat chloride together in combination, and fungicide Nexicor and fungicide application at heading stage had much significantly higher yield than check. And similarly, plant growth regulator applied at two node stage or heading stage, they also had significantly higher yield. Very similar result from another on-farm site of the same set of 5 treatments, and most of the treatments actually a heading stage application of Nexicor, and both stage 2 node and heading stage application of plant growth regulators have significantly higher yield than the check. So this is a third experiment conducted on farm with 3 different treatments, and those treatments applied at 2 node stage, plant growth regulator treatments, fungicide treatments, both had significantly higher yield than these are labeled little bit differently, higher yield than the check. So yeah, we all were affected by COVID-19, and because of that, we had little about six weeks of absence from the site, and we had to terminate greenhouse studies, and we had to also reduce outdoor activities with a lower staff capacity. We worked at about 20% capacity on site, so we are able to record fewer variables than we wish to. So concluding our work, in 2020, project activities are in progress to meet objectives. Change requests were submitted and already approved for the following years of activities. And the growth regulators and disease management treatments, especially in on-farm condition, they seem to be effective solutions for seed yield improvement for multiple years. And there are lots of opportunities for further studies, and we need to further analyze yield barriers and their economic significance. And we also have opportunity to conduct to study some post-harvest management, vertical tillage practices, and other agronomic manipulation for multi-year seed harvest of creeping red fescue dwarf grass. With that, I'd like to acknowledge funding and administrative support from Agriculture and Agri-Food Canada, Peace Region Forest Seed Association, Canadian Ornamental Horticulture Association, and collaborative support from Calvin Yoder. He used to be with Alberta Agriculture and Forestry. Recently, he is retired from there, and so he's associated with Peace Region Forest Seed Association, and other colleague Talon Gertier, Peace Region Forest Association, and our Beaver Lodge colleagues and technicians, Jennifer Otani, Henry, Rahman, Nobu, and Pat. With that, thank you so much for your patience and any questions you may have.